So when I was asked to do this, I wasn't sure really exactly what information I should be trying to give you, because you all know most of life is luck, and actually you can plan things as much as you want, and the way things turn out is often quite different. And so I think that's a good point to end the talk, because that's probably as much wisdom as I can give you. But I thought it might be interesting just to tell you about my journey through science, because I actually started life as a synthetic organic chemist. So often when I talk to undergraduates that work for me and they're really concerned and worried about their third-year project not being quite in the area that they want it to be, and really worried that this is going to set them sort of for a life in organic chemistry, for example, I sort of say to them, look, just look at me. I generally did my third-year project looking at platinum and palladium catalysis. And then I sort of decided that wasn't quite what I wanted to do. So when I was choosing a PhD, I went and talked to one of my tutors at Bristol, and I said, can I work with you? And he was doing organic chemistry. And so for the next 4 years of my life, I literally worked on a single reaction. So for those of you who aren't chemists, you can mix two things with some reagents, and you get a product, hopefully. And in this particular case, in the literature, it said that when you mix A and B, you get C. And my supervisor, Joe, was sure that actually you should be getting D. Except you weren't getting D, you were getting C. And so for 3 years, I did the same reaction, slightly changing conditions. Different base, different solvent, different temperature. What happens if I leave it a bit longer? Until I managed to get D, right? Until we got no C, and all we got was this compound D. And that was a pretty big thing, because no one else had managed to get D from A and B at that point. And so having sort of done that, and that's called methodology development in chemistry, I then did a postdoc, because I wanted to sort of move much more into biology. The compounds we were making, D, was an amino acid analog. And so we were thinking that it might be bioactive. We had collaborators. We were sending these compounds off. And you know what it's like. You're doing your own thing. Someone wants to collaborate with you. You get material, but it's not priority, right? So it sits in the freezer. And as the person who's generating that material, it's incredibly frustrating, because you're saying, I suffered to make this. I made this. What's happening with it? And so I sort of made a decision at that point that I would try and learn this other technique, biology, so that I would be in a position that I could make things and test things myself. So that would allow me to sort of close that loop and be able to immediately find out if something is good and if something has worked. And if it has, I could make analogs, test them, make them better, come back, etc., etc. Right? Now, there's always a necessity for collaborators. Right? You have to collaborate, because you're not going to be an expert at everything. But I think the more of it that you can keep in-house, it was certainly my view at the time, the more control you can have on it. So I made a decision after my PhD to make sure I'm tilting more towards training myself in biology. And so I did a postdoc at Sussex with a guy called Douglas Young. He's now retired. And that postdoc was, again, synthesis-based mostly. So I had to make this compound in 20-odd different steps that had a deuterium in one phase, and then another version of it where the deuterium was in the other phase, so we could figure out the kinetics of an enzyme to see whether it was this hydrogen that was being removed or this hydrogen. And as part of that, I was allowed to go over to biochemistry and learn how you would make a protein. So how you would make a protein in E. coli. I did a little bit of cloning. I did a little bit of protein expression. And so I was really starting to get a feel for this. And so I thought, okay, normally what you would do at that point is you get a job. Right? You've done a PhD, you've done a postdoc. 2 years, you get a job. And I thought, well, no, I'm not actually quite ready for a job. And at the end of my PhD, I was really desperate to go to America. I really wanted to experience US science, because, having been there and come back now, I can say this with confidence in that because there's more money over there, and because the funding, the funding schemes are different, the funding outlooks are different, the projects that are tackled, and it might just be just the general nature, the stereotype where they're a lot more bold and, you know, we can do it type, whereas we in Britain tend to be a lot more conservative and we don't like things that are very, very high risk. But the science is bigger, the problems tackled are bigger. And so it allows you to sort of have a different experience, right? And so I made a conscious decision after my PhD that I want to do biology and I want to do it in the States. I sent 100 letters. In those days, there were no emails. You actually literally had to write a letter, print it, put it in an envelope, pay for the postage and send it. And no one would actually accept me, right? So I was an unknown, the labs were full, you can come and work if you want for free, but we can't pay you. And so I was very, very lucky, and this is what I mean about life being more luck than plan, right? You end up where you are supposed to be. And so after my postdoc with Doug, towards the end, Doug started asking me, so what do you want to do, Ali? What do you want to do with yourself? Because I've always been incredibly hardworking and sort of I was always first in, last out. I knew my organic chemistry. I would really give the others in the lab a hard time, whether they knew their pKa's, whether they knew, because in organic chemistry, there's named reactions. You know, the person who first discovers this reaction is named after them. So it's a thing where you sort of ask someone, draw me the mechanism of the Wittig reaction, for example. And sort of the students, you know, I was really quizzing them on these sort of things, just to keep myself up to date as much as anything. So I was viewed as someone who was very serious and very good. And so Doug felt confident in recommending me to his friend Steve in the States. And Steve Benkovic, he's sort of, in the field, he's very big. He's world famous for enzymology. He's sort of runs one of these super groups at Penn State. He's been there all his life, and he's just a big name to work for. And sort of Doug sat me down and said, look, I know Steve. If you want, I can make a phone call and you can go to Steve. And I was like, oh, God, yeah, absolutely, you know. And this is just the difference between 2 years and, you know, having the patience to wait 2 years and working hard is literally phoned Steve up. Doug spoke to him. He called me into his office. 5-10 minute conversation on the phone, you know, at Steve telling me the work they do in their lab. And at the end of it says, so do you want to come? I was like, yeah, okay, see you in September. Just puts the phone down. It was that easy. I sent 100 letters, you know. And it was just that simple. And I think I can honestly say that changed my life. That there's very few points where you're at a fork and your life takes a turn. And at the time, the funny thing is, at the time, you don't know that this is what's happening. And it's certainly when you look back on it, you think, oh yeah, that was a fork. And it changed the direction of my life fully. Because, you know, Ali in an alternative life, I probably would have gone and worked for a chemistry company. I would be doing organic synthesis right now. And that would be me, right? And because of this shift, you know, Ali's now at Southampton, and, you know, we're doing chemistry, we're doing biology. And it's amazing to think that without being conscious about the decisions you're making, the magnitude of them, that these things happen. Anyway, so I went to Steve. I took a flight to the States, 2 suitcases packed, and I don't know. So when I was giving this, when I was talking to my wife yesterday, saying I'm just going to talk, I'm not going to have any slides. I just want to talk. You know, it's going to be a stand-up routine. And so she said, no, you must have some slides. I said, no, you don't need slides. Anyway, she convinced me I needed slides. So I had 3 slides. One of them was a map of Penn State to show you where it is exactly, because I bet no one knows where Penn State is, right? It's in the middle of Pennsylvania, in the middle of nowhere. There's 2 main streets. A quarter of it is the university, and the rest of it is just sort of people who work at the university or who work at the shops and offices and things that serve the university. And it's just the most wonderful place to be. They call it Happy Valley, and it's because you're so isolated from real life. Just things are happy there. I can't explain it, but if you ever get to go and work there, I highly recommend it. You know, it's the sort of place where your front door is always open, never locked. your keys. I remember going snowboarding one time, and my sister was visiting me, and I left my keys and my wallet in the car, just because I don't want it in my pocket. She's like, you can't do that. The car will be gone. You'll be fine. Sure enough, you come back, your wallet's there, your keys are there. Or if you have your bike in the back of your car, you just park up, go for food, come back, your bike's still there. It's that sort of place. It's just wonderful. Anyway, so I took a plane to DC, and then I remember coming out of DC airport, taking the local hopper plane, and it was one of these propeller planes. And it was, again, 2001, right? So the pilot had his door open, and he was sort of chatting to us, and look over there, that's such and such creek, and that's such and such national forest. And we landed, and I went to Steve, and initially I was hired to be a chemist. Right? So my skills were chemistry. Steve's lab was set up so that he had chemists, he had biologists, he had cell biologists, and he had molecular biologists. And so I was going to be one or two chemists. And again, this is where luck plays a part, right? So the project he'd hired me for, they were working on making inhibitors of this class of compounds called enzymes called methyltransferases. Because DNA is methylated, some DNA bases are methylated. This plays a really important role in us, in humans, in regulating gene expression. It also plays a really important role in prokaryotes, in bacteria, in their defense mechanism. Right? And these things are quite specific. So the theory was at the time that if you could inhibit methyltransferases, you would have really good antibiotics. And Steve's lab were making analogs of this compound called SAM, which is a substrate for the methyltransferase. The methyl group, which gets put on the DNA, comes from this molecule. And so if an enzyme binds this molecule, if you make something that looks like it, it can block the enzyme from working. And that's what they were trying to do. And the people on the project before me, they designed the molecule, and as part of their design step, they needed to purify it. And purifying it meant that they had to couple it to another compound just to make it a crystal so that they could purify it and then take that compound off and carry on changing this, right? So when the person who's testing these compounds comes, he picks up the wrong vial. Doesn't pick up the final compound, but picks up this intermediate compound that has the group on for the purification. Tests it, and it's by far the most active thing they've seen in their series. Right? And again, this is where I think luck plays a part in life, right? So picks up this compound, tests it, doesn't quite know what it is, thinks it's the end thing. They talk when the person comes back on Monday, realizes that they've taken an intermediate, and all of a sudden the project shifts. We're now no longer making analogs of the final compound, we're now making analogs of this intermediate compound. And so when I came in on the project, we were at that stage where they wanted me to develop these compounds a bit more. Around that time, so I was 11 days in, so I arrived on the 1st of September. It was Labor Day, so I spent a day doing nothing because everything was closed, and then I went to work the day after. And then around that time, 9/11 happened, sort of 10 days after. And then after that, you probably don't remember, but there were cases of anthrax being posted through the mail in the US. And so it was really the potential for harm from bioterrorism was really at the forefront of the government's mind. And having novel antibiotics was actually really something that was significant, right? Because these compounds contain boron, and there's very, very, very few examples of drugs on the market that have a boron in. Boron is just an atom, right? Most things are made of carbon, nitrogen, and oxygen. Sometimes they have fluorines in terms of pharmaceuticals, but to have a boron in is highly, highly unusual. And again, the thought was that by making this drug so unusual, it would be selective and it would be much more potent than anything the bacteria have seen before. So Steve got some money to start a company. They started the company in California because his collaborator at the time was at San Francisco. And so during my first year there, the work kind of got spun out into a company. They got 20 million from the defense agency to start a company called Anacor. Steve Baker, who was an old postdoc in Steve's lab, went to Leicester to start his own independent career, but sort of was unconvinced that that was right for him. came back to Anacor to lead it, to lead the chemistry side of things. And before long, the majority of the synthetic work of the project was actually being taken care of by the company and outsourced, right? So I wasn't needed anymore, right? So again, this wonderful thing about the US and wonderful thing about Steve is I went to him and I said, Steve, I know, my time here potentially on the project is coming to an end, but can I hang around? Can I hang around and just learn? Because, he had groups of biologists, he had people doing enzymology, and I didn't know much about any of that stuff, and I wanted to learn. And to Steve's credit, Ali, hang around. Sure. And this isn't hang around for free. Hang around and I'll keep paying you. And so it was just an incredible opportunity to learn. So the compound that we worked on were commercialized. The first compound to come out was called Tavassoli, right? So it was marketed as Keridin. And I think Pfizer bought, I know Pfizer bought the company. So Pfizer bought the company, I think it's about 400 million they paid for it. And the only compound they had on the market was Tavassoli. They had another compound coming through which was for psoriasis, which was actually really, really promising, which is why Pfizer bought them. Because, again, boron-based compound used to treat psoriasis, showing really promising preclinical data. And that was enough to convince Pfizer to take the compound off the market. Well, to take the company, not the compound off the market, to buy the company. And so I guess what I got to see from there, from a commercialization perspective, is just how things work in the States, how much money there is available. So to get 20 million to start up is not that unusual there, but I think it's fair to say it's highly unusual in the UK. And so I sort of now I can look back on it and have a good appreciation of the differences in the two systems and how, what we can aspire to, I think is the best way to put it. Because I think the difference where you have 5 years of running time, you have 20 million, you don't need to worry about money running out, you just have 5 years of running time, you can really push on a project and get it to where it needs to go commercially, versus the model where you're getting, 500,000, you've got 6 months running time, here's another 500,000, another few months of running time, here's another, do you know what I mean? So it makes a big difference to have that big upfront cash to let you run and really develop your ideas. So then, while I was hanging out at Penn State, I was talking to some of the other postdocs in there. And Alex, Sergey, Todd were already working on a project where they were using engineered bacteria to make libraries of cyclic peptides. So they would engineer E. coli so that each bacterium would make a different cyclic peptide. And the libraries you could generate were hundreds of million in size. So imagine a human making a library of 100 million things. It would take forever, right? It's not something that's really feasible. But you can engineer bacteria, so overnight they can make libraries of 100 million compounds. And you can also engineer them so that they will test those compounds against your disease of choice. So you can take proteins. So the first thing we did in my lab was we took proteins from human cells called Hypoxia-Inducible Factor, and these are the proteins that control how a cell responds when it runs out of oxygen. So this is critical when you're an embryo because you need the hypoxic areas in your developing body to be properly vasculated. So the vasculature, the neovascularization is signaled by hypoxia. In a tumor, it's also really important because tumors are very hypoxic. And they've shown that HIF, this protein, is 50-fold upregulated in most tumors. So if you could inhibit HIF, you would have a process for stopping tumors adapting to this hypoxic environment, and they would die. So the majority of tumors this would apply to. And so the challenge we set ourselves when I started the lab was to find an inhibitor of this protein-protein interaction, which was very tough at the time. But to go back to Alex, Todd, and Sergey and I, I was talking to them. I remember Sergey was instrumental in this. I was talking to him saying, look, I think I'm just going to go back because, Steve's being nice, he's paying me, but I don't have something I can sink my teeth into, and I'm done with this. I'd made the compound, it had gone to Anacor, they were dealing with stuff, that was out of my hands, and I wouldn't hear anything in terms of, here's some shares or here's money until 10 years later, which is how it always works, right? You do your bit for a project, it goes off, other people take it forwards, and they work hard, and then if it becomes something, you all share in the profits. And so I remember Sergey saying to me, it was 8 p.m., we used to work crazy hours, so I would come in super early and we would work till very, very late. And I even remember one year on Christmas Day going into work, and because my family were in the UK, so I decided to stay and push, and push on the project. And I remember walking across the sort of main street in State College and literally looking left and there was not a soul, and looking right and there was not a soul, and just going into lab and actually happily working because I cared about this stuff. I cared about my science, right? Anyway, it was about 8 p.m. Me and Sergey were chatting, and I said to Sergey, look, I think I'm going to go back. And again, another fork in the road that you just don't know is happening as it's happening. Sergey said, why don't you just come on this project with me and Alex and Todd? We're trying to do this thing. We could always use another pair of hands, and it would be a good way for you to learn cloning. You would learn, biophysics. You would learn all of these different things. And so I said, okay, let's check with Steve. If Steve is happy, I'll do this. And we talked to Steve, and Steve was, again, the lovely, lovely man that he is. He said, yeah, fine, just do it. And I think the key thing is just believing in people. I think as someone who's now in charge of people, I think that's the biggest lesson I've taken away from my time with Steve, is that you hire good people and you just believe in them and you leave them to get on with it. And I think that's sort of the biggest thing I've taken away from Steve is, just trust people, because they want to do well. People aren't with you. They're not giving you 2-3 years of your life to waste their time. They actually have aspirations and they want to do well. So if you trust them and if you give them everything they need to do well, 9 times out of 10 people will surprise you and they will do well. And that's a win-win for both the postdoc or the student and for the supervisor. Anyway, so it was all fresh. I still have my lab books from when I joined Sergey and Alex and Todd on the project, literally a recipe of how to do the simplest thing. So right now I look back and I laugh, how to make a gel, or how to sort of do a ligation. Instructions, detailed instructions that only someone who genuinely doesn't know what they're doing writes down. So I could go back and repeat this without bothering them every time I need to do a ligation or a PCR. Open the lid of the PCR machine by pressing. Silly things, right? But pretty soon I was up and running. And the target that Steve told me to go after was a protein called ATIC. And ATIC has a 5,000 angstrom interface when it's interacting. It's a homodimer, so it interacts with itself. And it controls the biosynthesis of purines. So that your DNA is made out of 4 things. 2 of those things are purines, 2 of them are pyrimidines. ATIC controls the biosynthesis. It's one of the key enzymes in the biosynthesis of purines in your body. And again, if you could break it apart, there were potential therapeutic consequences. But also, because it was the first time anyone was really trying this sort of thing, we wanted to see how far we could push it. If we put something that is really difficult into the system that we're building, would it work? And again, it took about 9 months to a year of solid work. And I remember when the positive result came through, it was a Friday. It was late. It was really late because everyone had gone. And normally we had, there were people who would work till crazy hours to the point that they had children. Their wife would bring the children in. They would have, half an hour with their children, and then the wife would take the children away, and they would keep working. Right? But this must have been really late because even Jun had gone home, and I was just like, well, if Jun's gone, it's bad. But I remember doing the key experiment of, does this whole system work? Right? Does this whole thing that I've spent almost a year building, has it worked? And it had. I still remember the graph, the inhibition graph on the screen, going up and working. And I wanted to tell somebody, right? So I was going around looking for some, because this is a big lab. There was 20, 25 people in the lab, 25 postdocs, and spread across the whole floor. And I just couldn't find anyone. And I had to wait till Monday to tell people that this thing had worked, right? But it was just the most amazing thing. And Steve, again, to his sort of credit, when I came to set up my own lab here, so I worked another year and a half with Steve in Steve's lab, and we developed more things and we did more things. But when I came to set up my own lab, he said, "You can take this with you," and again, sort of, you need to be lucky. Had I worked with someone who was a bit more selfish, had I worked in an environment where my colleagues were a bit more selfish, I might have never been invited onto that project. My supervisor might have never let me take this thing from his lab to my own lab, and he stops working on it. And even to this day, if he ever gets any requests for any material, he just forwards it to me and says, get this from Ali. You need these people in your life to help you get somewhere. You need people to help you. You need people to look after you. You need people to guide you. And Southampton, again, I mean, I've been here 11 years now. I came in 2006. And again, we've had that. Chemistry have always been wonderful. I went to them and I said, I need cell culture labs. Our project is at a point where I need to culture human cells, and I need to do some, and they gave me space to do this. I mean, space is at a premium, right? And for them to put aside, I mean, talking to my colleagues at other universities who were in similar situations, I don't want to use the word better is the wrong word, but universities that are ranked higher than us, and they were struggling to convince their department to give them the space, and their work was suffering. In chemistry, quick chat to the head of the department, yeah, this room's yours, that room's yours, get going. And again, you need this support, the sort of invisible support that happens behind the scene that you never see. And so that work has led to several first-in-class molecules coming out of our lab in Southampton. So when we started, inhibiting protein-protein interactions was a bit of there were very few examples of it. It wasn't something that people were necessarily convinced could be done. And many groups around the world thought this was wrong. And so we decided that, we all worked towards it. We all came at it from different angles, using different technologies and different methodologies, but we're at a place now where actually people are convinced, pharmaceutical companies are convinced that you can do this. The platform technology that we've established, this method of making hundreds of millions of compounds in E. coli and screening them against any target, we've worked with many companies in identifying first-in-class molecules that then get taken forward against a range of targets, not just cancer, HIV, antibacterial, and anti-obesity. So it's become quite a useful thing to have a platform that you can apply to anything, because you can apply it to anything. And we've developed the platform. The latest thing that I'm really excited about is we've moved out of E. coli and we've put the whole thing into femtoliter droplets. Again, chance meeting, there was a chap here called Martin Fischlechner, and just a chance conversation with him saying, look, can we do this? And him being kind enough to help and support us in an area that I had no idea what I was doing. And again, right, this seems to be a theme to my life, is I have no idea what I'm doing and people help me. So maybe I'm the wrong person to be giving this talk, but so Martin's kindness meant that we've now developed a system that is potentially truly world-beating, because the technical issues that were there with the previous system just are not there with this new system. And it has the potential to be sort of the leading method for generating and screening cyclic peptides against various targets. I think pretty much that's a good place to end it, and sort of let you ask me any questions that you might have. Yeah, thank you. Regarding funding of our current work, again, we're lucky in that because we have a platform that we can apply to a variety of things, then we can go to a variety of people for funding. So I've had funding from MRC, we've had funding from BBSRC, we've had funding from EPSRC. The biggest funder throughout my career has been Cancer Research UK, because I guess our biggest footprint has been in cancer research. So I was lucky that when I first started, I had a career development fellowship, which gave me 5 years of funding from Cancer Research UK. And then I was again very lucky in that they gave me a Programme Foundation Award, which is 6 years of funding to sort of develop stuff. So I think they have always been the bedrock, if that's the correct word, that has, really supported the lab very well. And that's people's money, right? And we feel a real sense of responsibility. And I sort of talk to my postdocs about this. I personally feel a very real sense of responsibility, and I make sure that the people working on these projects are aware, because that is money from people who've typically either had the disease or they have lost someone to the disease. And they're giving you that money with hope. And you have a real responsibility to look after that and not be wasteful with it, and to work your hardest with it. Yeah, I guess that's and industry. So on the individual target project basis, it comes through industry. So we've had 9 separate projects with industry based around the platform, where a company would come to us and say, we have this target, it's been through our internal screening, and it hasn't succeeded in finding an inhibitor. So we work with cyclic peptides. They are amino acids that are joined together in a ring, 6 of them typically. And typical drug discovery works with small molecules. And these are completely different chemical spaces. And while small molecules are good for targeting things with defined pockets, because there's a pocket for that molecule to go into, things like protein-protein interactions don't have defined pockets typically. So you're talking about large featureless surfaces which are not very amenable to a molecule binding in them. But the peptides do, because they're slightly bigger, and they can, because of the way our screen is set up, they can trap one of the proteins in a conformation where it doesn't interact anymore. And so because we're in a completely different chemical space and we have technology that isn't accessible, then I think that's again a bit of good fortune. So we're known for our work in the cyclic peptide field. We're known for having this platform, and people see the value of the cyclic peptides as potential starting point for therapeutic discovery. And so, people do come to us. It was a struggle at first. So I remember talking 2 years into my tenure at Southampton, talking to a company and sort of standing there presenting the system and saying, look, we've built this, we want to do this, these compounds are going to be useful, and essentially being laughed out of the room, right? To that same company coming back 4 years later when we had published the work on our HIF inhibitors. HIF has been known since 1995, and our compound is still the only molecule that specifically hits one isoform over the other, and only one of 2 compounds that can break apart HIF. The other one is non-specific. So once that comes out, I think people see the value of the platform and what it can deliver, which really changed the game for us. So I think most people are evidence-based, certainly most scientists are. And so once you can provide the evidence that something is successful, I think they buy it and they come on board, which is what's happened. The sad answer is no. And I tell you why no. I'm learning in different ways, and I'm learning different things. So I've got 2 little boys, and I learn from them. It's amazing the things outside of science that you're so engrossed in science that you have to push hard to become a professor. You can't take your foot off the pedal at all. There's always people who are willing to do more than you. There's always people who are smarter than you. There's always people who have more luck than you. So it's always been my attitude, that I have to work harder. I have to do more. I have to be better. I'm my own worst critic. I always have been. And when you're in the lab, you can learn every day, right? You can do things. And I still was in the lab till maybe 4 years ago. I still had my own bench space. I had my own pipettes. I would go in and make sure at least every day I'm doing an experiment, and the crazy stuff I would do myself. And then if it worked, I would give it to a student to try. And so at that point, I would say I was learning every day. But it just became too busy, you know. You get administrative duties, your teaching duties go up, there's more people in your group to look after. So it just became impossible to be in the lab. And so I think at that point, I just drive a desk at the moment. And I think what I'd really like to do is go back to the lab. And I think that's a real, I was thinking about this. It's funny you should ask that. I was thinking about this maybe a week ago, saying, right, I am going to block out, you know, 1 day a week where it's my lab day. And I'm going to refuse any meetings. I'm not going to do anything other than be in the lab. Because I think I'm here because I'm good at being in the lab, right? And I enjoy it. And so I need to make sure that that's an environment that I'm back. So I do feel like I'm still learning every day. But you do learn just different things, right? I don't think any of us ever stop learning. You make a mistake and you're kind of like, oh yeah, maybe I shouldn't do that. It depends. So there's different philosophies. So if you think about to what extent you make your own luck versus how much of it is the universe being kind, right? And it is just different philosophies, you know. It's Nietzschean versus, you know, yeah. So my personal belief is that it's a combination of the of both things. So had I not worked hard, had I not sacrificed a lot of things to dedicate myself to my science, I could have had all these kind, nice people around me helping me. I still wouldn't have got very far. And regardless of how hard I worked, had I not had these kind, nice people around me, I still wouldn't have gotten to where I got to. So I think it's a combination of the two. And I think the mistake a lot of people make when things don't work out is that they forget that it's not them fully, that actually there's a large element of luck in it, right? So, you know, if I write a grant application, it might be good, but if I'm unlucky and it goes to a reviewer who's in a bad mood, or if it goes to the funding agency and the person presenting that is not doing a great job, through no fault of my own, I may not get that grant. Equally, that grant could have been terrible to start with, and that could be why I didn't get it. But I think what most people forget is just the luck. And equally important to remember is when things do work out, I really strongly believe you shouldn't go around going, it's me, this is all me. Because it's not you, you've been lucky. And I think it's really important to remember that. And certainly that's my philosophy. Whether it's correct or not, I can't speak for it. But I think you make your luck by working hard. The rest you have to trust the universe to help you. Yeah. So that's a good question about the intellectual property So there's two approaches to that, right? So you can either patent everything, but that comes with cost, quite literally cost, right? So each patent you file, you have to pay a lawyer about £5,000 to write that patent. That patent is filed. It's good for about 18 months. Unless you pay another £5,000, it's not good anymore. That £5,000 buys you another 18 months. And then at that point, you either drop it or you file nationally. And that whole process costs about £50,000. So if you imagine at a university like this, you know, chemistry alone has about 45 academics. There's probably, and I'm pulling the numbers out of thin air, so I could be wrong, there's probably 500 odd academics here, right? If each of them wants to file a patent every 2 years and maintain that patent, just imagine the cost. You know, you would take the whole operating cost of the university and just be patenting people's patents. And then maybe one of them will pay off, maybe none of them will pay off. So depending on how much money each university has, they tend to take different approaches. So in the US, it was very much a case of, there is a filter. There are people who work in a tech transfer office, and they sort of make the decision on whether this work is worth patenting. Almost based on, partially based on the person's track record who's coming in. If I'm going in every day and saying, look, I want to patent this, you can't possibly have 20 things you want to patent, right? Yeah. So I think and the problem we have as academics is there seems to be kudos for these things, and there really shouldn't be, right? There seems to be a kudos to the number of patents you have. There seems to be kudos to, we're becoming much more metricized as a community, right? How many papers do you have? What impact factor journals are you publishing in? And that is, to my mind, that's just not what science is about. Science is about a person pursuing a problem, right? That's all science is about. I am going to give myself to find out more about this. And I kind of think science is sort of society's luxury. Society gets to a point where they have everything else they need pretty much, and they say, okay, we're going to give X amount of our joint income to these people to go and pursue problems and try and solve these things for us so we could have a better future. And I think that's how I view science is, we're paid by the public to solve problems. And it's irrelevant how many patents I have. We shouldn't even be judged on these metrics. It is possible that in the future the landscape will change, but my worry is that as a sector, we're actually going the opposite way. We're becoming much more metricized. Exercises like the REF, the Research Excellence Framework, exercises like the Teaching Excellence Framework, they are trying to measure something which by definition is unmeasurable, right? So if you when you're asked to review a paper for one of the research councils, I was asked to comment on this person's proposal and how it would pan out in the next 15 years. I was like, how do I know? How would anyone know how someone's idea will pan out in the next 15 years, how useful something would be in the next 15 years? The famous example is the laser, right? The laser was invented, just sat in a cupboard for 50 years until we now all have CD players and DVD players in our houses. And if you were going to judge whether the society needs a laser or not at that time, you would have probably said, no, who cares about this? What use is this thing? But that's what I mean about science being society's luxury. You just problem solve, and some of those solutions will be useful and some of them just won't be. And I think it needs a mindset change, not just in us as a community, but in people who fund us, people who judge us. We're increasingly managed. Universities didn't used to have nearly as much management as they do now. And with that management comes the managerial cultures, right? Where they try and metricize everything, they try and measure everything. How are you doing against how you were doing last year? So I think the whole sector is shifting in the wrong direction. I've been thinking about this a lot, to be honest with you, because I think partially it's how we fund science, as you say. I think here so even something simple like a grant not being renewable. So in the US, Steve still holds the same NIH grant. He's 75, 80, I think. He still holds the same NIH grant that he held when he first started his career, and he's just been renewing it. Right? So, I want to work on this problem. This is the progress I made. Give me some more money to work on it. Okay, have some more money. Okay, now 5 years later, I still want to keep working on this problem. Here's the progress I made. Give me some more money. And that simple mechanism of renewing a grant just doesn't exist here. You have to come up with a new idea. And I think we're partially very vulnerable to spin here, where suddenly everyone's doing synthetic biology. And, everyone's rearranging their research to fit in with this remit that some person in government has said is where we want to go. You go back nanotechnology. Just go back every 10 years and find a buzzword, and everyone's rearranging their research to fit in within that buzzword, to fit in with the research council. And actually the problem is that it's top down and it's not bottom up. Right? It should be us, the people on the ground doing the work, that feed up, that says, look, this is what we should be working on. Not the people at the top saying, this is the latest buzzword that we want to sell to government, So this is what you're working on. So I think those two little things, I think for me, is the sort of big difference. And there's money in the system here. There really is. There is lots and lots of money. But I think it's given out to what is perceived as safe pairs of hands. And actually, if you're going to do crazy stuff, right, it's going to be, the idea is going to seem crazy at the time. But again, it's that trust that I talked about earlier. You just have to trust people, good people, to do good work. You just have to trust them. And I sort of, in my mind, I keep going back and forth between, would it be better if we had a funding system where just everyone got similar amounts of money as academics, where we were given, the same amount of money each, and if you did well, there was money in the system that if you did well, it's like Formula One. It's not an equal playing field. They've all got different cars, and the people with more money and better cars, they will go faster and win. Right? And it's the same in academia. And I sometimes think it would be better if it was more like stock car racing, where everyone essentially has the same car, the same engine, so it's competitive and different people will be able to win. So even if you're in a small car, you could still win. So in terms of the future of the platform, the thing that I'm most excited about, so we never stopped tweaking, right? As a person, I'm a tool maker. I love making tools and letting people use them. And I should say the platform that we built at Penn State is fully open. So, we send it to anyone who asks for it. But I get emails from all around the world asking me to send them the plasmids and the strains, and we just send it out to people. Because for me, the more people that use it, the better. The more useful the thing that we built, me, Alex, and Sergey and Todd, is going to be. The future, so I am really genuinely excited about this droplet system, because knowing the field, there are companies in this field, in the cyclic peptide field. There's a company called PeptiDream that was started by a chap in Japan, and it's worth 3.5 billion, not million, pounds. There's Ra Pharmaceuticals that's worth 400 million. No, this is a million, 400 million. And so I think there's real opportunity to be competitive. And I think the technology that we've developed over the sort of last 3 years has the potential to be better than anything that's out there. So actually that's the thing I'm most excited by, is trying to do justice with that technology, in terms of trying to do the technology justice, trying to, we have something wonderful, but again, it's luck. Now, am I going to meet the right people who are going to help me push this forward? Or, you could have the best thing in the world, if no one is willing to buy into it and help you and push it forward, it's going to just languish, right? So if I am lucky, then hopefully in about 5-6 years' time, we would look back and say, look, Ali was right, this really was an important step change in the technology. Or if I'm wrong or unlucky, then we'll forget all about it, and I'll be here in 5 years' time telling you about something else I've built that might be important. Yeah, thank you for coming. Thank you all for coming.